Hello friends, welcome to my channel Easy Life Science. Here I am talking about some important questions, answers and their explanation which are frequently asked in a set life science exam. So please subscribe my channel Easy Life Science, like my video, share it to your friends and press the bell icon so you will get a notification of newly uploaded video. So let's start. Here starting from the question number 32. A combination of a heme with oxygen is called option A, that is oxyhemoglobin, is a correct answer. Oxyhemoglobin, it is a combination of a heme with oxygen. It is formed in a lungs when oxygen binds to a hemoglobin present in RBCs. It binds reversibly. The hemoglobin is a respiratory pigment present in RBCs. It is required for a transport of oxygen from lungs to a various tissue. So, option A is the correct option. Next question 33. Adult hemoglobin contains dash dash polypeptide chains. The option A, that is a 2 alpha and 2 gamma. The adult hemoglobin contains a 2 alpha and 2 beta, not A. The option B is a correct answer. Adult hemoglobin contains 2 alpha and 2 beta polypeptide chains. Hemoglobin consists of 4 subunits, each having one polypeptide chain and one heme groove. Hemoglobin has a quaternary structure. It is made up of two pairs of different proteins designated the alpha and beta chains. There are 141 amino acids in a alpha chain and 146 amino acids in a beta chain of a hemoglobin. Both a fetal and adult hemoglobin are quaternary proteins composed of 4 protein subunits. However, adult hemoglobin is composed of 2 alpha and 2 beta subunits, while fetal hemoglobin is composed of 2 alpha and 2 gamma subunits. If the question is asked a fetal hemoglobin contains, then option A will be the correct answer. But here is asked about adult hemoglobin, so option B is the correct option for question number 33. Next question 34. Each hemoglobin molecule carries dash dash number of O2 molecule. The option B, that is a 4. Each hemoglobin molecule carries a 4 number of oxygen molecules. Each hemoglobin molecule has a 4 binding site for oxygen. Thus, each hemoglobin tetramer can binds and carries a 4 oxygen molecules. Question number 35. Each gram percentage of a hemoglobin when fully saturated can carry dash dash ml. Option B, that is a 3.14 ml. Question number 36. Which of the following mineral is not involved in a formation of a hemoglobin? Option D is the correct option, that is magnesium is not involved in a formation of a hemoglobin. Question number 37. In a sickle cell anemia, the defect lies in which polypeptide? The option B is the correct option, that is a beta chain. A sickle cell anemia, it is a inherited blood disorder. Hemoglobin enables RBC to carry O2 from lungs throughout the body. In this sickle cell anemia, RBC become hard, looks like a English letter C, or looks like a farm tool, that is a sickle. Mutation in a hemoglobin beta gene can lead to a sickle cell anemia. The gene or a beta gene is found on a chromosome number 11. So the answer of question number 37 is a beta chain. Then question number 38. Average mean corpuscular diameter is dash dash micrometer. The option C, that is 7.3 micrometer. Average mean corpuscular diameter is 7.3 micrometer. Question number 39. Increase in RBC count beyond million per cubic millimeter is known as. It is known as a polycythemia. In a polycythemia, there is a increase in RBC count. It is also called as a blood cancer. It causes your bone marrow to make too many RBCs. These excess RBCs thickens your blood, slowing its flow, which may cause serious problems such as blood clot. And this polycythemia is because of a change in or a mutation in one gene, that is a JAK2 gene, JAK2, that is a JAK2 gene. The other option like anisocytosis. Anisocytosis means RBC are of unequal size. Normally, the RBC are of a similar size, but in anisocytosis, the RBC are of unequal size. The option B, that is a poikilocytosis. In this condition, the RBC are of unequal shape. And in leukocytosis, leukocytosis means increased level of leukocyte in a blood. Leukopenia, decreased level of leukocyte in a blood. So, the polycythemia is a correct answer for question number 39. Question number 40. During erythropoiesis, hemoglobin first appears in. During erythropoiesis, hemoglobin first appears in. Option B is intermediate normoblast. Hemoglobin appears first in intermediate normoblast. The erythropoiesis is a process in which there is a synthesis of erythrocytes or RBC, and RBC are developed from a erythropoietic stem cell. In early development of fetus, erythropoiesis takes place in a yolk sac, spleen and liver. After birth, all erythropoiesis occurs in a bone marrow. Number of stages are in a erythropoiesis, that is a pro-erythroblast, erythroblast, normoblast, reticulocyte and erythrocyte, that is a fully matured RBC. The erythropoiesis is stimulated by decreased oxygen in a circulation, which is detected by a kidneys, which then secrete the hormone erythropoietin. The erythropoietin secreted by a kidney leads to a process of erythropoiesis. So, during erythropoiesis, hemoglobin first appears in a intermediate normoblast stage. Option B is the correct answer. Question number 41. During hypoxia, kidney release. The kidney release renal erythropoietic factor. In a hypoxia, your body does not have enough oxygen. Without oxygen, your brain, liver and other organ can be damaged. At this condition, kidney release renal erythropoietic factor. So option B is the correct option for question number 41. Question number 42. Intrinsic factor is secreted by. The intrinsic factor is secreted by option C, that is parietal cells of a stomach. Intrinsic factor is a glycoprotein produced by a parietal cells of a stomach. It is necessary for a absorption of a vitamin B12 in a distal ileum of a small intestine. Whereas the chief cells of a stomach secretes a pepsinogen. It is a proenzyme or it is a zymogen. In a acid environment, it get converted into a protease, that is a pepsin. Pepsin is a protease kind of enzyme. Option D, that is a beta cell of pancreas. Beta cell of a pancreas secretes a hormone insulin in response to a high concentration of glucose in a blood, whereas the alpha cells of a pancreas secrete glucagons in response to a low concentration of a glucose. So the answer of question number 42 is option C, that is parietal cells of a stomach. Question number 43, which of the following extrinsic factor is required for maturation of RBC? For maturation of RBC, the extrinsic factor required are vitamin B12 and a folic acid. So option D is the correct answer for question number 43. Vitamin B12 and folic acid are essential for synthesis of DNA, cell division and maturation of RBCs. These are called as a extrinsic factor as it is obtained mostly from a diet. So next question, question number 44. Largest white blood corpuscle in a peripheral blood is? The largest WBC in a blood is the monocyte. They have large nucleus and generally these cells are multi-lobed. They make up about 7% of a total content of a WBC or leukocytes. The size of a monocyte is 15 to 18 micrometer in diameter. So the correct answer for question number 44 is a monocyte. The smallest lymphocyte, smallest WBC is a lymphocyte. The process, question number 45, the process by which WBCs squeeze through the pores in a capillary wall is? The answer of this question is diapedesis. Diapedesis is a squeezing movement of a WBCs from a capillary at site of infection. Generally, this process of a diapedesis is carried out by a monocytes. Other option like opsonization, it is a process by which opsonins bind to a surface of antigen so that the antigen will be readily identified and engulfed by a phagocytes for their destruction. The pinocytosis means the to drink. It is a process by which the cell takes the fluid along with dissolved small molecules, and this pinocytosis is an active transport process or it is an energy consuming process. So the answer for question number 45 is the diapedesis. Question number 46, smallest blood cell is? Option B is the correct option, is platelets. Platelets are a smallest blood cell. Platelets are also called as thrombocytes. They are associated with blood clotting and they have no nucleus. They are fragments of a cytoplasm which are derived from a megakaryocytes of a bone marrow. Normally, the platelets are 1,50,000 to 3,50,000 microliter per microliter of a blood, and they having very small in their size, up to a 20% less than that of the size of a RBC. So the answer of question number 46 is option B, that is platelets. Question number 47, commonest anemia in India is? Option C, that is iron deficiency anemia is a commonest anemia in India. Then question number 48, lysosomes are produced by? The lysosomes are produced by a Golgi apparatus. They are formed by a budding of a Golgi body. Mannose 6 phosphate receptor or a MRP receptor are the transmembrane glycoproteins that targets the enzyme to the lysosome. So, answer or option D is the correct answer for question number 48. Moving to the question number 49. Which of the following is not associated with a mitochondria? Option A, it is associated with ATP production. Mitochondria through a electron transport chain produces a ATP by a respiration. Cristae are foldings of inner mitochondrial membrane. Cristae increases the available surface area to enhance the productivity of a cellular respiration. The mitochondria are present in cells of eukaryotes like animal, plants, and fungi. It is double membrane structure. By aerobic respiration, ATP is generated, and it is called as a powerhouse of a cell. Means option A, option B, and option D. These are associated with the mitochondria, but the stroma is not associated with the mitochondria. Stroma is a fluid filling up the inner face of a chloroplast, which encircles the grana and a thylakoids. The light independent reactions or a night reactions takes place in a stroma. The stroma contains an enzyme that works with ATP and NADPH to fix a carbon to CO2 into a molecules like a glucose. So option C is not associated with a mitochondria. This is associated with a photosynthesis and a chloroplast. Question number 50. Which of the following statements are incorrect regarding to a ER? The adipose tissue has both smooth endoplasmic reticulum and rough endoplasmic reticulum. This is incorrect because the adipose tissue or a lipidous tissue has only the SER, that is a smooth endoplasmic reticulum. So option A is the correct answer. Option B, plasma cells has both RER and SER. Yes, it is correct. Hepatocytes has both RER and SER. It is also correct. RBC lack both RER and SER. It is also correct. So option A is a correct answer for question number 50. The term endoplasmic reticulum was coined by. The term endoplasmic reticulum was coined by a Porter. Option B. A Keith Porter visualized endoplasmic reticulum under electron microscopy. The endoplasmic reticulum is a phospholipid bilayer embedded with a proteins. Moving to the next question, question number 53. In rough endoplasmic reticulum, ribosomes are located on. Option A, that is a cytoplasmic site. In a rough endoplasmic reticulum, ribosomes are located on the cytoplasmic side. Question number 55. Which of the following statement were true regarding to a endoplasmic reticulum? Endoplasmic reticulum provides a structural framework to the cell. Yes, it is correct. Endoplasmic reticulum acts as an intracellular transporting system. It is also correct. Smooth endoplasmic reticulum is involved in a synthesis of lipid. It is also correct. So the option D is correct. All of the above. Protein glycosylation occurs in the. It occurs in a lumen of rough endoplasmic reticulum. Protein glycosylation is a process where the synthesized protein are stacked with another glycoproteins, or sugar molecules are attached to the protein through glycosidic bonds. It may occur in a Golgi apparatus and endoplasmic reticulum. N-linked glycosylation begins in a endoplasmic reticulum and a O-linked glycosylation appears in a Golgi apparatus. The lumen of rough endoplasmic reticulum is a site where there is a glycosylation occurs. Then question number 56. Golgi apparatus is absent in. Golgi apparatus is absent in option C, that is a bacteria and a cyanobacteria, that is a blue green algae. Option C is the correct answer. It is present in a higher plant cells. Yes, it is present in a yeast. So the option C is the correct option. Then moving to the question number 57. Which of the following statements are correct regarding a Golgi apparatus? Sorting and packaging. Yes. Exocytosis of melanin granules. Yes, it is correct. Exocytosis of thyroxine hormone. It is also correct. So option D is a correct answer for question number 57. The Golgi apparatus is responsible for transporting, modifying and packaging of proteins and lipids into a vesicle for delivery to targeted destinations in a cell. And the Golgi apparatus or a Golgi body, it is a membrane-bound organelle. It is made up of flattened stacked pouches called as a cisternae. So option D is the correct answer. The term Golgi apparatus was coined by. It is coined by a scientist Camillo Golgi. Option A is the correct answer. Option A is a correct answer for question number 58. Option B, that is a Robert Brown. The Robert Brown first time described the cell nucleus, and he is renowned for his Brownian movement and cytoplasmic streaming. He was a Scottish botanist. The option C, that is Robert Hooke. He observed a cork cell through his microscope. Robert Hooke was the first to visualize microorganisms. Option D, that is Benda. Carl Benda used first time used a crystal violet to observation of a cell as a specific stain, and Carl Benda was one of the microbiologists to use a microscope in studying the internal structure of cells. Option A is the correct answer for question number 58. The term Golgi apparatus was coined by, that is a Camillo Golgi. Then moving to the next question, question number 60. Question number 59. F0 and F1 particles are located on. The F0 and F1 particles are also called as elementary particles or oxysomes. They refers to a small round structures present within the folds of a cristae of inner mitochondrial membrane. They help in ATP production and oxidation. They are present towards the matrix site in a inner mitochondrial membrane. So the option B is a correct option for question number 59. Question number 60. In a mitochondria, cristae acts as a site for. In a mitochondria, the cristae acts a site for oxidation and reduction reactions. The mitochondria is made up of a double membrane, outer membrane and inner membrane. These are made up of a phospholipid membrane. So option D is the correct answer for question number 60. Question number 61. Mitochondrial inner membrane is rich in which phospholipid? The mitochondrial inner membrane is rich with a phospholipid, that is a cardiolipin. Option C is a correct answer. And cardiolipin is a hallmark lipid of a mitochondrial inner membrane. It is one of kind of a fat. Question number 62. Which of the following is not a function of mitochondria? Electron transport and associated ATP production. Yes, it is a function of mitochondria. Fatty acid breakdown. Yes, it is a function of mitochondria. non-severing thrombogenesis. Yes, it is a function of mitochondria, but the glycolysis and associated ATP production is not associated with mitochondria because the glycolysis or AMP pathway occurs in a cytoplasm. Question number 36, 63. Who coined the term mitochondria? Option B, that is a Carl Benda. First time term, give a term that is a mitochondria. Question number 64. Nucleus was first discovered by. Option C, that is Robert Brown. First time described a nucleus. He was a Scottish botanist. He is famous for his phenomenon that is a Brownian movement and a cytoplasmic streaming. Question number 65. Nucleus are absent in RBC and bacterium. RBC, sieve cell and bacterium. RBC only and none of these. Sieve cell, bacteria and RBCs are without a nucleus. So option B is a correct answer for question number 65. Question number 66. Nuclear membrane is a continuous connection with. The nuclear membrane is in a continuous connection with a rough endoplasmic reticulum. Question number 68. The DNA protein ratio in a chromatin is 1:1, that is option C. The nucleosome is a fundamental repeating subunit of a chromatin. Each nucleosome consists of a octameric core of histones. So option C is the correct answer for question number 68. The function of a nucleolus is. The primary function of a nucleolus is to produce and to assemble the cell's ribosomes. Also, the nucleolus is responsible for RNA, ribosomal RNA genes and their expression. So option D is a correct answer for question number 69. The basic protein of a nucleus are nucleohistones and nucleoprotamines are the basic proteins of a nucleus. Question number 71. Which of the following is called as perinuclear dense body? A lysosome is called as a perinuclear dense body. So thank you for watching my video. So please subscribe my channel Easy Life Science, like my video, share it to your friends, and don't forget to press the bell icon so you will get the notification of my newly uploaded video.